Good morning students. Today we are going to see about 2 marks and 3 mark questions in molecular genetics of 12th biozoology. Students, first of all you open your book, biozoology book 5th lesson, take the page number 64. First question, students, these questions are book inside and book back question. These are important questions. Study well, write 2-3 times, prepare well for the examination and get ready for the examination. First of all, 2 mark and 3 mark question we will see. First question, one gene, one enzyme hypothesis define. This is very important question. It is given in page number 64. In page number 64, right side one blue box is there, no? Within the box it is given one gene, one enzyme hypothesis. This hypothesis was proposed by Beadle and Tatum, George Beadle and Tatum in 1940s in Neurospora crassa. It's a fungi. Neurospora crassa is a fungi. That means mold, one of the mold, fungus. George Beadle and Tatum done this experiment in Neurospora crassa and find out the hypothesis, one gene, one enzyme hypothesis. And this is for 1 mark question. It is very important. And the definition for one gene, one enzyme hypothesis is given in 4th line. Students, open the bracket. One gene, one enzyme hypothesis, which states that one gene controls the production of one enzyme. Close the bracket. Next, second question. Define one gene, one polypeptide hypothesis. This is also very important question. This page number given in page number 64. In the second paragraph, that is in the blue box only. Second paragraph, last 3 lines. This hypothesis states that one gene controls the production of only one polypeptide chain of an enzyme molecule. Bracket close. Close the bracket. Next, third question. Define transformation. Page number 65. It is given in page number 65. Define transformation. That is one box is given, no? Above the box, the last 4 lines. In the last paragraph, from the last 4 lines. That is, this phenomenon, transformation, means by which DNA isolated from one type of cell when introduced into another type is able to retain some of the properties of the host strain is referred to as transformation. Next question, define gene. Page number 64. Students, in 5.1, gene as the functional unit of inheritance heading, it is given, no, that in that topic, second paragraph, third line. Gene is defined as discrete particles that follow Mendelian rules of inheritance, occupy a definite locus in the chromosome, and are responsible for the specific phenotypic character, and are responsible for the expression of specific phenotypic character. Close the bracket. Next, fifth question. What are the properties of genes? Page number 64. Page number 64, below the definition gene, no, that is given 6 points. Out of the 6 points, for 5 mark question you should write 5 points, and for 6 points, and for 3 mark question 3 points, for 2 mark question 2 points, like that you write. Number of genes in each organism is more than the number of chromosome. Point number 1. Several genes are located in the same chromosome, and the space of the gene in a chromosome is called locus. Second point, genes are arranged in linear order like beads on a string. Third point, gene occupies a specific position called locus. That also I said in the first point. Gene may exist in several alternate forms called alleles. Gene may undergo sudden change in position and composition called mutation. Genes are capable of self-duplication, producing their own copies. Close the bracket. Next question. Question number 6. Define ribozyme. Define ribozyme. Page number 68. In your book, take page number 68. Define ribozyme. In page number 68, RNA world, it is given no 5.5 RNA world. In that paragraph, in the right side, come to 10th line. RNA has the ability to act as both genetic material and catalyst. Open the bracket. RNA has the ability to act both as genetic material and catalyst. There are several biochemical reactions in living system that are catalyzed by RNA. This catalytic RNA is known as ribozyme. Catalytic RNA is known as ribozyme. Then next, 7th question. DNA is more stable. Give reason. Page number 69. 69. DNA is more stable. Give reason. Page number 69. Page number 69. Stability. They are given the second paragraph. Stability. DNA is very, very stable. Why? Give reason. Page number 69. In that, to come to the 10th line, that is, in DNA, the two strands are being complementary. DNA is made up of two complementary strands. Close the bracket. Then second line, that is second point, that is last of that stability paragraph. Presence of thymine instead of uracil in DNA confers additional stability to DNA. Presence of thymine in DNA. Instead of thymine, in RNA it is given, uracil is taken that part, taken the place of thymine. But in DNA, it is occupied by thymine. Because of the presence of thymine, it is more stable. Second point. Third point, DNA being information storage, it is given third point, no. And that last line come. DNA being more stable stores of genetic information and RNA transfer genetic information. DNA store genetic information, RNA transfer genetic information. That is no need. That 2 points is enough. One is DNA is made up of two strands of DNA are complementary. Then second is DNA is made up of thymine. DNA consists of thymine. Due to the presence of thymine, it is very, very stable. Then next question, 8th question. Why virus having RNA genome mutate and evolve faster? Why virus having RNA genome mutate and evolve faster? For this, answer is in page number 69. Page number 69. RNA virus, that is 69. Last paragraph, variation through mutation. DNA and RNA are able to mutate, but RNA being unstable mutate faster rate. Then second point is, you come to the stability passage, second paragraph. In that, below the DNA complementary strand, no, below that next point, bracket, open the bracket, 2-OH group present at every nucleotide in RNA is reactive group that makes RNA liable and easily degradable. So RNA mutate very fast. Thus the virus having RNA genome with a shorter lifespan can mutate and evolve faster. Then next question, 9th question. What are the 4 histone protein found in DNA nucleosome? Page number 70. Answer is in page number 70. Third paragraph. Left side, third paragraph. That is third paragraph, fifth line. The 4 histone proteins H2A, H2B, H3 and H4 are organized to unite to form 8 molecules called histone octamer. That is protein. That histone is, DNA consists of 4 types of protein, histone protein, and the 4 histone proteins are H2A, H2B, H3, H4. Then next question, differentiate euchromatin, heterochromatin. Page number 70. Euchromatin, heterochromatin is given page number 70. In page number 70, 5.8, DNA replication above that paragraph, that is page number 70, first paragraph, right side first paragraph, come to the last 6 lines. In a typical nucleus, some region of chromatin are loosely packed and are referred to as euchromatin. The chromatin that is tightly packed is called heterochromatin. Euchromatin is transcriptionally active and heterochromatin is transcriptionally inactive. Second point. Differentiate, it is given 2 points. First point is for euchromatin in a typical nucleus, that is in a particular nucleus, some region of chromatins are loosely packed and are referred as euchromatin, and it is euchromatin is very active, transcriptionally active. And heterochromatin means the chromatin that is tightly packed is called heterochromatin and it is transcriptionally inactive. Second point. Next question. Define transcription, page number 73. Students, take page number 73. Page number 73, 5.9, transcription. In that second paragraph, open the bracket. The process of copying genetic information from one strand of DNA into RNA is termed transcription, and the process takes place in presence of DNA-dependent RNA polymerase. Close the bracket. Next question. 12th question. What are the 3 regions in a transcription unit? What are the 3 regions in a transcription unit? Page number 73. For this, answer is page number 73. Page number 73, transcription unit and genes, given one paragraph topic 5.9.1. In that, transcription unit in DNA is defined by 3 regions. One is promoter, next is structural gene, and terminator. Close the bracket. Then next question. Both strands of DNA are not copied during transcription. Why? Both strands of DNA are not copied during transcription. Why? For this, answer is given in page number 74. In page number 74, page number, both strands of DNA are not copied during transcription. Oh, sorry, page number 73. In page number 73, transcription 5.9, transcription topic is there, no? In that, come to third paragraph. Come to the third paragraph, right side. Third paragraph, both strands of DNA are not copied during transcription for 2 reasons. One is, if both the strands act as a template, they would code for RNA with a different sequence. This in turn would code for protein with a different amino acid sequence. This would result in one segment of DNA coding for 2 different proteins, hence complicate the genetic information transfer machinery. Second point is, if 2 RNA molecules were produced simultaneously, double-stranded RNA complementary to each other would be formed. This would prevent RNA from being translated into protein. Close the bracket. And then next question, define reverse transcription, give example. Define reverse transcription, give example. For this, answer is in page number reverse transcription. Reverse transcription is in page number 73. It is given in page number 73. Reverse transcription is given in page number 73. Note it. that is transcription, that second paragraph last. In some retrovirus, example is retrovirus. Give example I gave. For that example, retrovirus. Retrovirus means RNA having virus is called retrovirus. In some retrovirus that contain RNA as the genetic material, the flow of information is reversed. RNA synthesizes DNA by reverse transcription, then transcribed into mRNA by transcription, and then into protein by translation. Close the bracket. Next question. Define Goldberg-Hoeknes box or Tata box. Page number 73. Given in page number 73, last line in eukaryote, that is in the topic 5.9.1 transcription unit and gene, in that last line. In eukaryote, the promoter has ATG rich region. AT means adenine thymine. Adenine thymine rich region called TATA. That means thymine, adenine box. And this is called Goldberg-Hoeknes box. And in prokaryote, this region is called Pripnow box. Close the bracket. Then next question, 16th question. Differentiate template strand and coding strand. Page number 74. Students, this question also very important. Template strand and coding strand. Differentiate. Sometimes it may ask like define template strand and define coding strand. And it is also they will ask like differentiate template strand and coding strand. Template strand. For this, page number 74, second paragraph, left side. DNA-dependent RNA polymerase catalyzes the polymerization in only one direction. The strand that has the polarity 3' to 5' acts as a template and it is called template strand. The next is, on the other hand, which has the polarity 5' to 3' has the sequence same as RNA and is displaced during transcription. This strand is called coding strand. Tabular column, you should write this. Then next one is monocistronic mRNA, polycistronic mRNA. Differentiate. This is also very important question. Page number 74. Monocistronic mRNA, polycistronic mRNA. In eukaryote, example for monocistronic mRNA is in eukaryote. In eukaryote, each mRNA carries only a single gene and encodes information for only a single protein. If a single gene encodes a single protein, then it is called monocistronic mRNA. In prokaryote means bacteria-like organism, cluster of related genes known as operon found next to each other on the chromosome are transcribed together to give a single mRNA, and hence they are called polycistronic mRNA. Cluster of genes known as operon. Cluster of genes is otherwise called operon, found next to each other on the chromosome, that is found on the chromosome, are transcribed together to give a single mRNA, then it is called polycistronic mRNA. Then next question, in transcription, what are the function of 3 RNAs? Function of RNA. It is given in page number 74. Students, please take page number 74 and 5.9.2 process of transcription heading. In that, there are 3 RNA, mRNA, tRNA, rRNA. In prokaryote, 3 major RNAs are needed for transcription. First of all, mRNA provides the template. Function of mRNA is it provides template for protein synthesis. Then tRNA bring amino acid and read the genetic code. Function of tRNA is bringing amino acid and read the genetic code. Then function of rRNA, it is structural and catalytic role during translation. It acts as a structural and catalytic role during translation. The next question, Capping and tailing differentiate. 19th question. Capping and tailing. Page number 75. Students, take your page number 75 in your book. Capping and tailing. Page number 75, right side. First paragraph, last. You come to the 11th, 12th line. That is HNRNA. Heteronucleotide RNA. HNRNA undergoes additional processing called as capping and tailing. In capping, an unusual nucleotide methylguanosine triphosphate is added at the 5' end, that is called capping, whereas adenylate residue are added at the 3' end is called tailing. This is capping and tailing. Capping means unusual nucleotide, that is methylguanosine triphosphate is added to the 5' end of the chain, then it is called capping, whereas adenylate residue are added at the 3' end of the transcription DNA material, then it is called tailing. The next question is define splicing. Page number 75. Splicing. Students, above that capping and tailing, that is from the in the page number 75, 10th line, right side 10th line, first paragraph 10th line. In eukaryotes, the monocystronic structural genes have interrupted coding sequence known as exon and non-coding sequence called introns. The introns are removed by the process called splicing. Removal of introns from monocystronic structural gene is called splicing. The next question, define genetic code. Page number 75. Students, page number 75, genetic code is given in the 5.10. Page number 76, 5.10, genetic code, second paragraph. Genetic code is the sequence relationship between nucleotide in gene and the amino acid in the protein they encode. There are 64 possible triplet and 61 of them are used to represent amino acids. Next question, define non-ambiguous code. Define non-ambiguous code. Page number 77. That is above the tabular column, fourth point. Right side, non-ambiguous code means that one codon will code for only one amino acid. If one codon denotes one amino acid, that is called non-ambiguous code. The next question, nonsense code define. Page number 78, 79. Last line. UAA, that means uracil adenine, uracil adenine guanine, UAG. UAA, UAG and UGA, uracil guanine adenine. Codes are designated as termination codon and they are called nonsense codon. These three codons, UAA, UAG, UGA. These three codes are terminator codon and they are called termination codon and they are called nonsense codon. The next question, wobble hypothesis. Page number 78. Wobble hypothesis. Page number 78. In page number 78, it is given in the blue box, wobble hypothesis, from the second line. tRNA anticodon has the ability to wobble at its 5' end by pairing with even non-complementary base of mRNA codon. Close the bracket. This is wobble hypothesis. According to the hypothesis, in codon-anticodon pairing, the third base may not be complementary. The third base of the codon is called vowel base and this position is called vowel position. Close the bracket. The next question, define translation. Page number 80. Students, take page number 80, translation. 5.12. Translation refers to the process of polymerization of amino acid to form polypeptide chain. Translation refers to the process of polymerization of amino acid to form polypeptide chain is called translation. Then next, define Shine-Talcano sequence. Shine-Talcano sequence. Page number 81. In page number 81, last 4 lines. The ribosome binding site is called Shine-Talcano sequence, SD sequence. The sequence base pair with the region of the 16SR RNA of the small ribosomal subunit facilitating initiation. That is from above line. The 5' end of mRNA of prokaryote has a special sequence which precedes the initial AUG start codon of mRNA. This ribosome binding site is called Shine-Talcano sequence or SD sequence. Close the bracket. The next is, write down the application of DNA fingerprinting. Page number 88. Students, take page number 88. Application of DNA fingerprinting. Forensic analysis, pedigree analysis, conservation of wildlife and anthropological studies. Only you should write heading for 2 mark or 3 mark question. For 5 marks only detailed answer should be given. You should write. It's given in page number 88. It's also 5 mark question. If it is asked in 5 marks, then you should write in detail. If it is in 2 mark or 3 mark question, you should write only heading. Forensic analysis, application of DNA fingerprinting. It is used in forensic analysis, it is used in pedigree analysis and conservation of wildlife analysis, and it is also used in anthropological studies. The next question, define VNTR and its significance. Page number 87. VNTR and its significance. VNTR means variable number tandem repeats. VNTR, the full form of VNTR is variable number tandem repeats. It is given in page number 87, first line only. In DNA fingerprinting, short repetitive nucleotide sequences are specific for a person. These nucleotide sequences are called variable number tandem repeats, and the significance is the VNTR of 2% generally show variation and are useful as genetic markers. VNTR is used as genetic markers because any 2% of the VNTR may vary. VNTR means variable number tandem repeats. In DNA fingerprinting, short repetitive nucleotide sequence of a person is called VNTR. The next question, what are the challenges of mapping of human chromosome? What are the challenges in mapping human chromosome? Challenges in mapping human chromosome, it is given in page number 86. Very important question. Once genetic sequence become easier to determine, some people may attempt to use this information for profit or for political power. Second point is, insurance company may refuse to insure people at a genetic risk. For disease like genetic disease, insurance company won't allow any insurance. So it will be a very challenge to the people. The next point is, another fear of the attempt are being made to breed out certain genetic genes of people from the human population in order to create perfect race. So according to Darwin, some species are superstitious species like that. So people may develop only perfect race of people. That also create ethical problem in humanity. The next question is, define pharmacogenomics. Page number 86. It is given in the box. Did you know? Pharmacogenomics is the study of how genes affect a person's response to drugs. Close the bracket. Students, so today we go through molecular genetics, 2 mark and 3 mark questions in molecular genetics of biozoology. Students, these questions are very, very important. Please study these questions and write down 2-3 times. Then only you will get good practice. For the exam purpose, it will come like a flow if you are thorough with the subject. So please study this and get ready for the examination. Thank you. God bless you.